And one minute. Welcome எல்லாமே சொல்லி தானே போறேன். ஸ்டார்ட் பண்ணட்டும். எவ்வளோ ஸ்டூடண்ட்ஸ் இருக்கு? சவுண்டு எல்லாமே போயிட்டுருக்கா. ஸ்டார்ட் பண்ணியே போதும். ஓகே. பண்ணட்டும். ஹலோ ஹலோ ஓகே. Hi all future doctors, welcome to the question discussion of Kerala NEET Academy. Okay, today we are going to discuss about 4 important chapters of first year botany. Okay, so we start with the first chapter. It's a very simple chapter and you will get 2 to 3 questions from this chapter for your coming NEET exam. And the chapter is plant growth and development. So moving into the first question. Here you can see that the first question, growth can be measured in various ways. Which of these can be used as a parameter to measure growth? So this is the first question we are going to discuss. Okay. So can you answer the question? First question. Question related topic is growth in plant growth and development. So okay. So the right answer is, here the right answer is option 4. All the above. Why it is all the above? So we understand growth. What do you mean by growth? Growth is a fundamental and also it is the conspicuous characters of living. So in the case of growth, growth can be measured by using different parameters. So we understand from plant growth and development, growth has got 2 important characters. First one is growth is measurable. Growth enna panna mudiyum, measure panna mudiyum. அப்படி growth measure பண்ணானாயிட்டு இப்போ நம்ம இது வந்து ஒரு செல்லு, இந்த செல்லு டிவைடு பண்ணிட்டு புதுசாக 2 செல்லு ஃபார்ம் பண்ணும், இந்த செல்லு எகெயின் டிவைடு பண்ணும், இப்படி புதுசாக செல்லு ஃபார்ம் பண்ணும். Here you can see that here at the beginning you can see only one cell and then it undergo division to form many cells. Here you can see that the number of cell increases. That means cell number increases. Here cell number increases. Okay, and then this is a pistil and after pollination you know that when pollen grain falls on the pistil, this pollen grain undergo growth as pollen tube. So in the case of here the pollen tube is growing. Here pollen tube is showing the growth, but it is in which pattern? Lengthwise. Pollen tube development is in lengthwise. And here there is a small leaf. After some time this leaf gets expand. That means area of the leaf is increases. This is an apple fruit. After some times its size gets increases. Here the size increases. That means growth of a plant body can be measured in different ways. That means in the case of cell we calculate the growth by counting the cell number. In the case of pollen grain we can calculate the growth by measuring the length. In the case of leaf we can use the area, and also in the case of fruit we can use the size. Okay, and also another parameter and that is the weight. You can also use the weight as well. Okay, we can calculate the weight. Weight, we all know that there are 2 types, fresh weight and dry weight. Okay, that means, so from this first question we understand that growth is a fundamental characteristic fundamental characteristics of living, and also this growth is measurable, and this growth can be measured by using different parameters, and the parameters are cell number, length, area, size, and also weight. Okay, so this is a point related to the question number 1. Okay, so we completed the question number 1, then we move on to the question number 2. Okay, and this is the question: the type of growth in which parent cell undergo mitosis to form 2 daughter cells, in which one matures and other is divided. So we understand what is growth. Okay, growth per unit time is called growth rate, and this question is from the topic growth rate. This question is from the topic growth rate. So can you answer the question? Okay. Okay, so we are going to discuss the question. So it's a type of growth. We understand that growth per unit time. Growth per unit time is called what? Growth rate. So when you consider the growth rate, you can see that there are 2 type of growth rate are there. And there, this is one growth. Here there is an individual cell, it undergo divisions. Okay, here there is one cell, okay, this is one cell, it undergo divisions. 2 new cells are there, and the division is here is mitosis. Division here is mitosis. Here one cell undergo differentiation and the other cell continue to divide. Okay, and also here it will also undergo mitosis, and this cell undergo differentiation. This will continue to divide again. This is a one type of growth rate, and in another type of growth rate, this individual cell undergo mitosis. As a result of this, 2 new cells are there, and each cell will undergo division, mitosis again. So this kind of growth rate is called arithmetic growth rate, and this kind of growth rate is called geometric growth rate. Okay, so in our question, the question is: one parent cell undergo mitosis to form 2 daughter cell. One parent cell, this is the parent cell, which will undergo mitosis to form 2 daughter cells. In that, one matures and other divide. Here you can see that this will matures. That means this will undergo differentiation, and this will start divide. So this kind of division can be seen in which growth rate? This can be seen in arithmetic growth rate. Okay. So we come, so the right answer is option 1. Right answer is option 1. Okay. Right answer is option 1. So next question, the ability of a plant to follow different pathways and produces different structures in response to environment or phases is known as. So it's a very important topic from the plant growth and development. Can you answer the question? Okay, so the right answer is, here the right answer is plasticity. Here the right answer is plasticity. So what do you mean by plasticity? Okay, plasticity, right answer is plasticity. Okay. So in NCERT, they have given two examples, larkspur and buttercup. Both these plants shows two different type of leaves. So in the case of larkspur, we all know that in its juvenile phase, this is the juvenile phase leaf, and also when we look at the adult phase, the leaf pattern is entirely different in adult phase. So plant shows two different type of leaf in response to the phases of their life. Okay, and in the case of buttercup, we understand that in its terrestrial habitat, leaf is like this, but in the aquatic habitat, leaf may divided and forms like this. That means based on the environmental or based on the habitat, it produces two type of leaf. This is called heterophily. This is called heterophily, and this heterophily is example for what? Plasticity. Heterophily is example for plasticity. Okay, clear? So the ability of a plant to follow different pathways to produce different structures, in response to environment or phases is called what? Is called plasticity, and heterophily is an example for plasticity. Okay, clear? So next question from plant growth and development is match the following question. It's a very familiar question for you. It is repeated in many times. Okay, so match the following question. In column A you can see the scientist name, and next column you can see the hormone. Okay, can you answer the question? Okay. Okay, so we are going for the discussion of the answer. So we all know that cousins discovered which plant hormone? Ethylene. Discovered which plant hormone? Ethylene. Okay. Miller and Scooch, we understand that it is cytokinins. Okay. Then E. Crosa, we know that it was gibberellins. Then there is F.W. Wendt, he discovered, he isolated auxins. So F.W. Wendt is related to auxins. So the right answer here is option 2. Option 2 is the right answer here. Okay. So next question is also a match the following question from the chapter plant growth and development. In column 1 you can see the functions of, in column 1 you can see the hormone names, plant hormones or phytohormones, and in column 2 you can see the functions. Okay, can you answer the question? Okay, can you answer the question? Okay. Okay, then we are going for the discussion of the answer. So column 1, auxins. We all know that auxin, one of the important function of auxin is the initiation of root or root initiation in stem cuttings. So we understand this. Okay, then gibberellins, one of the important function of gibberellins here is gibberellins helps in breaking the seed dormancy. Okay, seed germination is a function of gibberellins. So it will break seed dormancy. Then cytokinins and also ethylene, formation of abscission. Okay, then breaking of seed dormants. Then cytokinins nama ellarkume theriyum cytokinins function. Chloroplast development and chlorophyll synthesis. Cytokinin chloroplast development and chlorophyll synthesis. Okay, then ethylene, நம்ம எல்லாருக்குமே தெரியும் ethylene function இது, it is inducing the fruit ripening. So these are the functions of for plant hormones, auxins, gibberellins, cytokinins and ethylene. So this is the final question for the chapter plant growth and development. Okay. Okay, now we are going to discuss about the next chapter, and the next chapter is anatomy of flowering plants, and the next chapter is anatomy of flowering plants. So the first question, it's an image based question, and you will get many questions from this chapter as image based questions. நிறைய questions in the chapter in image based ஆனது. So first question, okay, can you answer the question? Okay, question number 6. See the following figures and identify the type of simple tissue marked by alphabet. Here you can see A, B, C. Can you answer the question? A, B, C. Okay. Okay, let's check the question. Okay, option A. Option A. This is a structure of sieve tube. Companion cells எல்லாமே இதுக்குள்ள இருக்கு. Okay, so we all know that in the case of plants there are two type of tissues are there. They are simple tissue and permanent tissue, and this question comes from the permanent tissue. Permanent tissue, you know that there are two types, simple permanent tissue and complex permanent tissue. In the case of complex permanent tissue, there are xylem and phloem. So this is coming from phloem. So as we know, phloem is composed of 4 different type of cells. They are sieve tube, companion cells, phloem parenchyma, like that. Okay, so question number 6, A, B and C. Okay, A is what? A is sieve tube. A is sieve tube, and B is phloem parenchyma, and C is the cell that control the function of sieve tube, and the cell is companion cells. So you can say that sieve tube, then there is phloem parenchyma, and then there is companion cells. Okay, 3 important type of cells in phloem complex tissue. Okay. Next question, the arrangement of xylem in stem is. Arrangement of xylem in stem is option 1 end arch, then option 2 X arch, option 3 mesarch, option 4 both 1 and 2. Can you answer the question? Can you answer the question? Okay. Okay, so the right answer is option 1 end arch. The right answer is option 1 end arch. When you are looking at the cross section of stem, you can see that the outermost layer is epidermis. Within that epidermis, you can see the cortex. Within that cortex, you can see the endodermis. Within the endodermis, you can see the pericycle. Within that pericycle, you can see the vascular bundles. These are the vascular bundles there. At the center, there is a homogeneous tissue which is called pith. Center pith. Okay, so when you are looking at the vascular tissue, you can see that this is the xylem bundles. Okay, so you can see that this is the small xylem which is called protoxylem. This is the large xylem which is called meta. And this is called metaxylem. See that protoxylem is actually facing towards what? Pith. So when the protoxylem is facing towards the pith, it is called endarch xylem. So the right answer is option 1 end arch. Right answer is option 1 end arch. Okay. Next question. Bulliform or motor cells are present in option 1 dicot stem, option 2 upper epidermis of dicot leaves, option 3 lower epidermis of monocot, option 4 upper epidermis of monocot leaf. Okay, I will wait, you can answer the question. Okay. Bulliform cells or motor cells. Okay, the right answer is okay, very good. The right answer is upper epidermis of monocot leaves. Okay, right answer is upper epidermis of monocot leaf. So when you look at the leaf, you can see that there are two type of epidermis are there. They are upper epidermis and lower epidermis. So in grasses, so namma vandu grass paathittunda. In grasses, upper epidermis consists of certain specialized cells. Certain specialized cells. Upper epidermis namma paathittunda adukulla ennadu paaka mudiyum konjam specialized cells paaka mudiyum. These specialized cells, upper epidermis is also called adaxial epidermis. This is the upper epidermis and this is the lower epidermis. When we look at the upper epidermis, we can see that there are certain specialized cells. These specialized cells seen at the upper epidermis is called bulliform cells. These cells are called bulliform cells. And the important thing about this bulliform cell is these cells are empty and these cells are colorless. Okay, these cells are empty and these cells are colorless and they are specific from their other upper epidermis cells. Okay, clear? And when you look at the function of this bulliform cells perform certain important functions. So one important thing is when they absorb water, bulliform cells vandu water absorb panna time il when they absorb water, the bulliform cells becomes turgid. They becomes turgid. When they becomes turgid, when this bulliform cells absorb water, ivaru vandu water absorb pannumbam ennadaidum turgid. So that what happens, leaf surface and the time il leaf surface enna pannidum, leaf surface becomes exposed. Okay, then when bulliform cells lose water, ivarkullayinu water veliyil poyidunna time il the condition is called flaccid. And the time il leaf enna pannidum, leaf curls, leaf surface curls. Okay, so this is the function related to bulliform cells, and you should understand that bulliform cells are present in the upper epidermis. They are present in upper epidermis. They are colorless empty cells. When they absorb water, they becomes turgid. Leaf surface is exposed. When they lose water, leaf becomes flaccid. So that leaf surface curls. Okay, leaf surface curls. Okay. So next question, ground tissue include. Next question is ground tissue include. Okay. Option 1, all the tissues internal to endodermis. Then next option, all the tissues external to endodermis. Then all tissues except epidermis and vascular bodies, and then there is epidermis and cortex. Okay, can you answer? Okay, 4 options. Okay. Okay. Okay, so the right answer is option 3. Right answer is option 3. All the tissues except epidermis and vascular bodies or vascular tissue. So in the case of plants, we know that there are 3 type tissue systems are there. So in the case of plants, cells organize or cells aggregate to form tissues. Okay, and tissues perform all functions in plants. So that you can see there are 3 type tissue system in plants. They are epidermal tissue system, ground tissue system and vascular tissue system. Okay. So ground tissue system. So question is about ground tissue system. Ground tissue system involves, when we look at the epidermal tissue system, you can see that epidermal outgrowth, okay, epidermal outgrowth, then there is stomatal apparatus. They contribute to epidermal tissue system. And vascular tissue system, you all know that there is xylem and phloem. They makes vascular tissue system. Appo idukapram plant kulla irukkunna all tissues ennathane ground tissue system. That means the ground tissue system, when you look at the ground tissue system, and the ground tissue system il namma paathittunda ennathakku irukku, cortex irukku, adukulla enadhu irukku, pericycle irukku, then there is pith. Okay, nama NCERT paathittunda, in the NCERT il namma last part il enadhu koduthirukku, ground tissue system koduthirukku. Summary paatha nama NCERT ila topic la summary il koduthirukku ground tissue system definition and the ground tissue system ude differentiation endha edathirukku summary il koduthirukku. Okay, next question. The following type of vascular bundles are present in. Following type of vascular bundles are present in. Okay. Okay. So option 1, monocot stem and leaf, then dicot root, monocot leaf respectively, then root, monocot stem and leaf, then there is dicot stem respectively, then root, stem, leaf respectively, stem, root, leaf respectively. Can you answer the question? Question number 10 and the last question from the chapter Anatomy of Flowering Plants. Okay. Okay. Right answer is option 2. Right answer is option 2. Okay. So when you look at the root, you can see that xylem, phloem like that. That means xylem and phloem are seen in different radius. So this is seen in where? This is seen in root. So in the case of stem, when you look at the stem, you can see that this is a vascular bundle. This is phloem and then here there is the xylem. So xylem and phloem are seen in a same radius. And also in the case of dicot root, dicot stem, you can see that in between primary xylem and primary phloem there is a specialized tissue, and this specialized tissue seen in between xylem and phloem is called cambium. So this is seen in where? So this is seen in dicot stem. So this kind of vascular bundle is a characteristics of dicot stem, and this is seen in stem and this is seen in root. So the right answer is option 2. Okay. So we completed plant growth questions and also we completed the questions from Anatomy of Flowering Plants. Okay. So moving on to the next chapter, and the chapter is very important, and the chapter is photosynthesis in higher plants. So first question. So the first question, The experiment material used by Van Niel to prove that oxygen comes out from water was studied on. Okay, it's a very important experiment. It's a very historical experiment. Okay, can you answer? So question actually about a Van Niel experimental material enna question. Okay, can you answer the question? Okay, so option is okay, very good. The option is option 3, purple and green sulfur bacteria, because before this Van Niel's experiment, scientists believed that oxygen evolved during photosynthesis comes from the splitting of carbon dioxide, not comes from the water. So that Van Niel conducted experiment using bacteria like a purple bacteria and green sulfur bacteria and demonstrated that oxygen evolved during photosynthesis comes from where? It comes from water, not from carbon dioxide. Okay. So carbon dioxide, water gives carbohydrate and then the releasing of oxygen. This was the equation given in NCERT, first equation given in NCERT. Okay, so previously it was believed that the carbon dioxide splits to form carbon and oxygen is released. This carbon will combine with water to form carbohydrate. Okay, this was the belief before the time of Van Niel. Okay, Van Niel demonstrated that carbon dioxide. Okay, this was the equation given in NCERT, and this 2A is, that means Van Niel experiment பண்ணிய டைமில் Van Niel என்ன பண்ணிட்டு இருக்கு, instead of hydrogen donor, here the hydrogen donor is a water, in his experiment instead of water as the hydrogen donor, he used what? He used H2S. Okay, and the released gas is sulfur. Okay, so he understand that it was not the carbon dioxide that splits and gives you the oxygen, it was water that splits and releases the oxygen during photosynthesis. So this experiment was done by Van Niel. Okay, next question, splitting of water is related with okay, option 1 photosystem 1, option 2 photosystem 2, then option 3 both 1 and 2, then option 4 cyclic phosphorylation. Can you answer the question? Okay, so எல்லாருமே இங்க பாரு, நம்ம இந்த क्वेश्चन டீடைல்டா ஸ்டடி பண்ணான் தானே போடுது. நம்ம எல்லாருக்குமே தெரியும் photosynthesisல லைட் ரியாக்ஷன் நம்ம பார்த்துடுமா, அது வந்து சைக்ளிக்கும் இருக்கு நான் சைக்ளிக்கும் இருக்கு. நம்ம நான் சைக்ளிக் ஃபோட்டோஃபாஸ்போரிலேஷன் பார்த்தா நம்ம எல்லாருக்குமே தெரியும், இப்படி ரெண்டு ஃபோட்டோ சிஸ்டம் இருக்குது, ஃபோட்டோ சிஸ்டம் செகண்ட் இருக்குது, அந்த மாதிரி என்னதும் இருக்குது, ஃபோட்டோ சிஸ்டம் ஒன்னும் இருக்குது. எப்போ ரெண்டு ஃபோட்டோ சிஸ்டம் இருக்குது, ஃபோட்டோ சிஸ்டம் 2 and also photosystem second. So this is the light harvesting complex of photosystem second, and this is the light harvesting complex of photosystem 1. We know that both this photosystem will accept light energy. When they accept light, what happens? They get excited and they releases the electrons. And these electrons are accepted by electron acceptor. Electrons are accepted by electron acceptor. Okay. From the light harvesting complex of 1, these excited electrons are accepted by electron carrier and they never return to their original site and they move towards the light harvesting complex of photosystem 1. Okay. So that electrons from this electron acceptor now move towards the NADP. This NADP will also accept H plus ions and getting changes into NADPH H plus. Okay. So that there is a deficiency of electron and this deficiency of electron can be covered by the splitting of water. Water gets splits into and then there is oxygen is evolved and the electron released by the splitting of water now reaches the light harvesting complex of photosystem 2. So that we can say that splitting of water, this splitting of water is related with what? Splitting of water is related with what? Photosystem 2. So the right answer is option 2. Okay. So next question, which was the first carbon dioxide fixation product formed in the Kelvin experiment using radioactive labeled carbon 14 in green algae? So question number 13 from photosynthesis. Okay, can you answer? So and the question is coming from Kelvin cycle. Can you answer the question? Okay. So the right answer is very good. Option 2. A 3-carbon organic compound, which was the first carbon dioxide fixation product. So we all know that in Kelvin cycle, RUBP. In Kelvin cycle, what? RUBP enna pannidum? RUBP will combine with the carbon dioxide. RUBP enna pannidum? Combine with what? Carbon dioxide. RUBP carbon dioxide ma combine panniya, it will releases a 3-carbon compound which is called 3-PGA. 3-phosphoglycerate, 3-PGA. So ನಮ್ಮ question என்னது? question இப்படி தானே வந்தது. question எப்படி? which was the first carbon dioxide fixation product. This process is called carbon dioxide fixation, which was the first carbon dioxide fixation product. Okay, carbon dioxide fixation is the question. So carbon dioxide fixation, this is the carbon dioxide fixation. This process is called carboxylation. So the right answer is option 3, a 3-carbon organic compound. And you should understand that RUBP is a 5-carbon compound. Here it consists of only 1 carbon atom. So how many molecule 3-PGA is formed? 2 molecule 3-PGA is formed during the carbon dioxide fixation. So this is 3-PGA is the first carbon dioxide fixation product. Okay. So next question, question number 14. Total requirement of ATP, then NADPH for each molecule of carbon dioxide fixed and reduced in photosynthesis in Kelvin cycle. 4 options are there. Can you answer the question? Can you answer the question? Can you answer the question? Okay, very good. The right answer is option 3. The right answer is option 3. 3 ATP and 2 NADPH. Okay, so let's check the question. Okay, so we understand that Calvin cycle, RUBP combines with carbon dioxide in the presence of an enzyme called, and then can you answer the enzyme? The enzyme is Rubisco, to form a 3-carbon compound called 3-PGA. This process is called carboxylation. This process is called carboxylation. And this 3-PGA undergo a process called reduction. Reduction resulting in the formation of triose phosphate. Reduction resulting in the formation of what? Triose phosphate. During this process, 2 molecule ATP and 2 molecule NADPH are utilized. As a result of this, what? ATP getting converted into ADP plus inorganic phosphate, and NADPH is getting converted into NADP. So how many molecule ATP is utilized here in the reduction? 2 molecule ATP and 2 molecule NADPH. And the triose phosphate, some part is getting converted into sucrose and the remaining part gets converted into RUBP. So you see that RUBP is again produced. So this step is called regeneration. This step is called regeneration. In the step, will also 1 molecule of ATP is utilized. So it is getting converted into ADP plus inorganic phosphate. So in total, how many ATP molecules are utilized? 2 plus 1. 3 molecule ATP, and how many molecule NADPH is utilized? 2 molecule NADPH is utilized. So the right answer is 3 molecule ATP. So inge paru 3 molecule ATP and 2 molecule NADPH. Okay. Next question. Photorespiration is a wasteful process because photorespiration, நம்ம எல்லாருக்குமே தெரியும் இந்த சாப்டர்ல இம்பார்ட்டண்டா இருக்கிற ஒரு டேர்ம் தான் என்னது? photorespiration. அது வந்து என்னது? ஒரு wasteful process னு தான சொல்லியிருந்தது. Why? Okay, அதுதானே options. First option, there is no synthesis of sugars. Second option, there is no synthesis of ATP or NADPH. Third option, there is no synthesis of phosphoglycerate. Option 4, both 1 and 2. Can you answer? Okay, can you answer? எல்லாருமே ட்ரை பண்ணு. Okay. Can you answer? Okay. So, okay, very good. The right answer is option 4. Right answer another option 4, both 1 and 2. The reason is there is no synthesis of sugar and there is no synthesis of ATP or NADPH. Instead, it utilizes ATP. So that photorespiration is a wasteful process. So what do you mean by photorespiration? இங்க பாரு, நம்ம எல்லாருக்குமே தெரியும் RUBP can combine with both oxygen and carbon dioxide. So RUBP combines with oxygen instead of carbon dioxide. Okay, and it produces 1 molecule of phosphoglycerate and 1 molecule of phosphoglycolate. Okay, and this phosphoglycolate releases carbon dioxide by utilizing what? By utilizing ATP. See that it utilizes ATP. There is no synthesis of sugars. Okay, so that's the reason why it is called photorespiration. It is called photorespiration. നമ്മൾ എല്ലാവർക്കുമേ തരും റെസ്പിറേഷനുള്ള ടൈമിൽ ഓക്സിജനെ എടുത്തിടും കാർബൺ ഡൈ ഓക്സൈഡിനെ താനേ റിലീസ് പണിത് இந்த இடத்தும் அப்படி தானே ஆக்சிஜனை கம்பைன் பண்ணிட்டு கார்பன் டை ஆக்சைடு ரிலீஸ் பண்ணி so respiration during what time? During photosynthesis. So the term is photorespiration. Okay. Okay, so moving from photosynthesis to respiration. So first question is the net gain of ATP in anaerobic respiration. So question is net gain of ATP in anaerobic respiration. Okay, can you answer option 1, 2, then option 2, 4, then option 3, 36, option 4, there is 32. Can you answer? Net gain of ATP in anaerobic respiration. Okay. Okay, right answer is very good. Option 1. Net gain of ATP during anaerobic respiration is option 1, it is 2. Okay, so நம்ம எல்லாருக்குமே தெரியும் when glucose is respired it will undergo a process called glycolysis. It will undergo a process called glycolysis. As a result of this, 2 molecules of pyruvic acid is produced. 2 molecules of pyruvic acid is formed. Pyruvic acid வந்து 3 carbon compound, glucose வந்து 6 carbon compound. Okay, so during this process நம்ம எல்லாருக்குமே தெரியும் direct ATP synthesis இருக்கு NADP NA. Okay, இந்த இடத்துல நம்ம பார்த்துட்டு இருந்தா டைரக்டா ATP ப்ரொடியூஸ் பண்ணிடுது. இது வந்து 10 steps இருக்குன process தானே glycolysis. Okay, and also okay, இது வந்து 10 steps இருக்குன process தானே. Okay, இந்த இடத்து pyruvic acid ப்ரொடியூஸ் பண்ணியதுக்கு அப்புறம் இந்த pyruvic acidக்கு 3 ஃபேட் இருக்குன்னு தானே NCERT ல கொடுத்துட்டு இருக்குது. அதுக்குள்ள இருக்குன ஒரு ஃபேட் தானே என்னது anaerobic respiration. Anaerobic respiration. இது வந்து anaerobic respirationல போயிடுന്ന டைமில் தானே என்னது நெட் கெயின் ஆஃப் ATP எவ்வளோ வந்திருந்தது 2 ATP. ஓகே. Okay, next question, very simple question. Cellular respiration is carried out by. Question is cellular respiration is carried out by. You can see that cell organelles names are there. Ribosome irukku, chloroplast is there, then mitochondria, and then there is Golgi bodies. We all know that cellular respiration. So cellular respiration irukuna cell organelle. The cell organelle which participate in cellular respiration is mitochondria. So the right answer is here is mitochondria. Mitochondria is the site for which kind of respiration? It is a site for aerobic respiration. It is the site for what? Aerobic respiration. Okay. Okay, question number 17, right answer is so mitochondria. Okay, next question, which of the following forms the connecting link between glycolysis and Krebs cycle? Glycolysis, Krebs cycle. Okay, so option 1 glucose, option 2 ethyl alcohol, option 3 lactic acid, Then option 4, acetyl coenzyme. So can you answer? Can you answer? So all the future doctors, can you answer? Okay, right answer is acetyl coenzymes. Option 4, acetyl coenzymes. Okay. So நம்ம எல்லாருக்குமே தெரியும் glucose undergo a process called glycolysis, resulting in the formation of 2 molecules of pyruvic acid. Okay, if the condition is aerobic, this pyruvic acid enter into this 2 molecule of pyruvic acid enter into a cycle, and the cycle is called Krebs cycle or citric acid cycle, or you can use the term TCA cycle, or you can use the term TCA cycle. Okay, and the pyruvic acid formed during the glycolysis is not the product for what? Not the substrate for citric acid cycle. So the pyruvic acid formed during the glycolysis is now converted into another substrate, and the substrate is called acetyl coenzyme. This process is called link reaction. This process is called link reaction, or you can use the term oxidative decarboxylation. Okay, link reaction. Question is, which of the following forms a connecting link? Question is about a connecting link between glycolysis and Krebs cycle. So we understand that. So acetyl coenzyme is the substrate that connects glycolysis with a citric acid cycle, or TCA cycle, or which is also known as Krebs cycle, because it was discovered by Hans Krebs. Okay. Next question, glycolysis gives rise. Glycolysis gives rise. Option, 4 options are there. Can you answer? So ellarume answer pannu. Can you answer? Question number 19. Glycolysis give rise. Glycolysis give rise. Glycolysis give rise. Okay, very good. The right answer is option 3. Right answer is option 3. Glycolysis gives 2 direct ATP, 2 NADH and 2 molecules of pyruvate. Okay. So let's check the answer. So we all know that glycolysis, glucose is converted into pyruvic acid. Okay, glucose is a 6-carbon compound. Glucose is a 6-carbon compound. In the 6-carbon in the glucose convert panni enadakidum pyruvic acid convert pannidum. Pyruvic acid vandu 3-carbon. So from 1 molecule of glucose, 2 molecules of pyruvic acid is produced. How many molecule pyruvic acid is produced? 2 molecules pyruvic acid is produced. Pyruvic acid is also known as pyruvate. Okay, and this is not a single step process. It involves 10 steps. Okay, in this 10 steps we understand that there is a net gain of 2 ATP. How many ATP is gained? There is a direct net gain of 2 ATP, and also how many NADH is produced? NADH. How many NADH is produced? 2 NADH is produced. So the right answer is 2 ATP, 2 NADH, and then there is 2 molecules of pyruvic acid. Okay. Then RQ is less than 1/4. last question. RQ is less than 1/4. Okay, inga paru options paathudu proteins and organic acid, fatty acid and organic acid, fatty acid and proteins, then proteins and carbohydrate. Okay, can you answer the question? Okay. Okay, so the right answer is, okay, very good. The right answer is option 3, fatty acids and proteins. Fatty acids and protein. So what do you mean by RQ? What do you mean by RQ? RQ நம்ம எல்லாருக்குமே தெரியும் RQ. RQ என்னதுதானே? The volume of carbon dioxide released to the volume of O2 consumed. It is called RQ. அதுதானே என்னது வந்திருக்குது? RQ ன்னு நம்ம சொல்லிடும். Okay, RQ நம்ம எல்லாருக்குமே தெரியும் கார்போஹைட்ரேட் யூஸ் பண்ணுன டைமில், கார்போஹைட்ரேட் யூஸ் பண்ணுன டைமில் இப்போ நம்ம எல்லாருக்கும் இது வந்து கார்போஹைட்ரேட் C6H12O6 plus 6O2 gives us 6CO2 plus 6H2 plus energy. So இந்த இடத்துல நம்ம RQ பார்த்துட்டு இருந்தா 6 molecule carbon dioxide divided by 6O2. நம்ம எல்லாருக்குமே தெரியும் RQ என்னது 1. அப்போ நம்ம கார்போஹைட்ரேட் யூஸ் பண்ணிட்டு இருந்தா, கார்போஹைட்ரேட் யூஸ் பண்ணிடும்போ RQ என்னது 1. இங்கே கொஸ்டின் என்னது? RQ is less than 1/4. RQ is less than 1/4. இங்கே பாரு, நம்ம ஃபேட் யூஸ் பண்ணும்போது என்சிஆர்டியில் ஒரு எக்ஸாம்பிள் கொடுத்துட்டு இருக்கு. அது வந்து ட்ரைபால்மெட்டின். நம்ம கார்பன், ஹைட்ரஜன், ஆக்சிஜன். ஓகே, நம்ம ஃபேட்டில் என்சிஆர்டியில் கொடுத்துட்டு இருக்குது ட்ரைபால்மெட்டின் தானே. (145O2) gives us 102 carbon dioxide plus 98H2 plus energy. ഇതാണ് നമ്മ NCERT-ൽ ഇരിക്കുന്ന ഇക്വേഷൻ. സോ വോളിയം ഓഫ് കാർബൺ ഡയോക്സൈഡ് evolved 102 divided by 145O2. RQ നമ്മ എല്ലാവർക്കുമേ தெரியும். RQ എന്നതാണ് NCERT-ൽ കൊടുത്തിരിക്കുന്നത്. RQ இந்த ടൈമിൽ കൊടുത്തിട്ടിരിക്കുന്നത് എന്നതാണ് less than 1 தானே. NCERT-ൽ കൊടുത്തിരിക്കുന്ന വാല്യൂ சொல்ல முடியுமா? NCERT-ൽ കൊടുത്തിരിക്കുന്ന വാല്യൂ சொல்ல முடியுமா? RQ എவ்வளோ தானே NCERT-ൽ കൊടുത്തിരിക്കുന്നത്? 0.9 தானே കൊടുത്തിരിക്കുന്നത്. ഓക്കെ, 0.7 தானே കൊടുത്തിരിക്കുന്നത്. ഓക്കെ, അപ്പോൾ RQ എന്നത് தானே ലെസ് ദാൻ 1 தானே കൊടുത്തിരിക്കുന്നത്. ഇനി വന്ന് RQ എന്നത് ഇരിക്കുന്ന ടൈമിൽ നമ്മ പ്രോട്ടീൻ യൂസ് பண்ணுന്ന ടൈമിൽ RQ എന്നത് தானே நம்ம அது பார்த்துறாണ്ട് NCERT-ൽ ഡയറക്റ്റ് അവർ എന്നത് കൊടുക്കാതെ ഇക്വേഷൻസ് ഒന്നുമേ കൊടുക്കാതെ, ബട്ട് അത് സ്റ്റഡി பண்ணால் நம்ம ഒരു ഇക്വേഷൻ സ്റ്റഡി பண்ணுന്നത് പ്രോട്ടീൻ. ഓക്കെ ஓகே, இது வந்து நம்ம ப்ரோட்டீன். ஓகே, ப்ளஸ் நம்ம ஆக்சிஜன் தானே யூஸ் பண்ணுது. ஆக்சிஜன் நம்ம யூஸ் பண்ணி how many molecules oxygen? 6 molecule oxygen. அப்போ என்னது கிடச்சிட்டு இருக்கு? 5 molecule கார்பன் டை ஆக்சைடு கிடச்சிட்டு இருக்கு. அந்த மாதிரி 5 molecule. ஓகே, தென் கொஞ்சம் எனர்ஜி ப்ளஸ் என்னது NH2 இப்படி தானே கிடச்சது. அப்போ நம்ம RQ பார்த்துட்டு இருந்தால் எப்படி கிடைக்கும்? RQ volume of carbon dioxide divided by oxygen. ஓகே, RQ எவ்வளோ கிடைக்கும்? less than 1. RQ less than 1. அப்போ RQ less than 1 இருக்கிறது நம்ம பார்த்தா அது என்னതൊക്കെ தானே இருக்கிறது? ഫാറ്റി ആസിഡും പ്രോട്ടീനും. ஓகே, ഫാറ്റി ആസിഡും പ്രോട്ടീനും இது. இந்த ஈக்குவேஷன் NCERT-ல் கிடையாது. நம்ம அண்டர்ஸ்டாண்ட் பண்ணனாட்டு தானே இந்த ஈக்குவேஷன் ரைட் பண்ணி. ஓகே, so we discussed 5 questions from the 4 chapters. I think it is very clear for you. Okay, and then thank you.